of you here. Um, we are a group of people promoting the idea of public health at UT. And so we have two people today, and I'm pleased to introduce Shelley Payne, who's director of the LaMontagne Center for Infectious Disease in the Department of Molecular Biosciences at UT. And at LaMontagne, microbiologists and immunologists study the life cycles of bacteria and viruses, how they become pathogenic, and how our immune systems combat those infections. And they are working to develop new strategies to prevent and treat infectious diseases. And we are really pleased. Shelley is a longtime campus leader in many ways. And so we're pleased to have you here, Shelley, and I will turn it over to you.
Great, thanks. And I will tell you at the outset, you are all not microbiologists, and we have a really bad habit of falling into jargon. So please cough, raise your hand, something when I start doing that. I get excited talking about the stuff and forget that not everybody is immersed in the minutiae of this as I am. But the kind of goal of my lab is providing basic information that other people can take and hopefully develop into interventions or treatments for diseases, infectious diseases. And we've really focused on bacterial pathogens. And the ones that my lab has worked on are ones associated with diarrheal diseases. I'm glad this is not a lunch talk. And these are primarily ones that cause epidemics and pandemics. So Vibrio cholera, which causes cholera, and Shigella species, which cause bacillary dysentery. And can everybody see the screen okay?
Yep.
Okay. Okay, so let me go on to the next. And so today I want to talk about some studies that we're doing on Shigella species. And this is a pathogen that is largely problematic in areas where sanitation and access to clean water are problematic. So you see a lot of it in sub-Saharan Africa, in Asia, in areas of South and Central America, less so in the areas where you do have access to good sanitation. But it still causes occasional epidemics and certainly a lot of sporadic cases. But I think you may have noticed in the news there's an outbreak of Shigella going on in the Portland area right now. And one of the problems with this one is Shigella is one of the most, if not the most, infectious of the bacterial pathogens. You probably only need somewhere in the order of 10 to 100 of the bacteria to cause infection. And to kind of put this into perspective, if you were to look at a person who had an active case of Shigella and you measured the number of bacteria in the diarrhea, the fecal contents that were coming out, it's going to be on the order of 10 to the 9th per mil or per gram. So billions. And so it only takes a minuscule amount of that to transmit to another person. So even if you have great sanitation and things, people can actually spread it to person during an outbreak. It's also become a problem to treat because of high levels of antibiotic resistance. So it's now on the WHO list of high priority pathogens. Another reason that we really want to understand this disease better is that often these cases are in young children. And children who have severe cases of Shigella can develop stunting and developmental delays. So this can have a lifelong impact on these people. This one also has been found to frequently co-occur with viral infections. And about 30% of the Shigella infections, they will also find that there are intestinal viruses, rotavirus, norovirus, enteroviruses can co-occur with this. And so one of the questions that we've been wanting to ask is, what happens when people get infected with both a bacterial and a viral pathogen? Do they interfere with each other? Do they actually make the other disease worse? Or really looking at more at the molecular level, what's going on in this case. One of the things we know about any of the diarrheal illnesses is that right after acquiring the pathogen, you have an innate immune response. And this is your body's typical response when you've encountered anything that shouldn't be there, and it's not specific. So it may deal more with certain classes of pathogens, bacteria or viruses or fungi, but it's your body's first attempt to try to get rid of whatever this is. With many bacteria, including Shigella, one of the early responses is called a pro-inflammatory response. So you get this strong inflammation. And I think any of you that have ever had a splinter that got a little infected, you know that you get pain, swelling, it feels hot, you get pus. And this is all of this signaling going on to say, let's contain whatever this is, bring in white cells, and try and clean it up before it can cause infection or severe disease. At the same time, you're beginning to make that the really effective immune response, which is going to be the antibodies and T cells, but that takes time. And it's this first response that we were looking at. So with Shigella, it's an inflammatory response. And in your gut, it's much like if you get a splinter, you get these areas of dead tissue, you get pus, bleeding into the area, and it's quite a strong inflammatory response. With viruses, those also use signaling to start an innate immune response. And one of the things that is most evident in that is an interferon response. So interferon, this class of signaling molecules that kind of says, we don't know what this is, but it's probably some type of virus. So let's beef up the antiviral responses. And these interferons are divided into several classes, one of which, interferon gamma, it's produced during some bacterial infections. But interferon alpha and beta, these signals are called the type 1 interferon response, and that's the one I'm going to talk about today. So again, in response to your body seeing these viruses, they produce this signal, the interferon, which then talks to all the other cells in the area and lets them know there's a virus coming in. So we looked at the type 1 interferon response to see what would happen with Shigella if you had a co-infecting virus and you were getting interferon, type 1 interferon produced. So Shigella comes in through the fecal-oral route. You get a little bit of the organisms in, and unlike many of the intestinal pathogens, this one infects in the colon. So it goes through the small intestine and then really gets in your colonic epithelium. And you have cells in the colon called M cells, which are designed to kind of sample the environment. They take things in, pass it off to your immune cells for processing and to kind of say, if we see this again, we need to be ready. But some pathogens, including Shigella, kind of hijack this. They come in through those cells, which are actively taking them up. And then when they get taken up by the immune cells, they kill those cells. So they're able to evade getting cleared. And then they come in through the back door and get into your intestinal epithelial cells and actually replicate inside your cells. So in many ways, these look like some of the viruses. They get in, hijack the host cell, replicate. And so now your immune system can't really see them. So the antibody and T cell responses have difficulty handling these. They spread into adjacent cells directly. They don't go outside. They go directly into the next cell and set up this replication. And as they do this, these cells begin to die. your body responds with this very strong inflammatory response, and you get fever, the diarrhea, all of this, and a lot of the symptoms are due to your own body responding to these bugs. We don't have a good animal model for Shigella, so what we use are human cells in culture. And there are two assays that I'm going to mention on this. In the first one, we look at how well the bacteria can infect the host cells. So we add the bacteria to the host cells, let them sit for a little while. Some of them will get in. We then add antibiotics to the outside, so we kill off any that didn't actually get in. We let them grow, and then we can stain and look at how well the cells got infected. So here you can see all of these little purple rods. Where'd my pointer go? Okay. These are all bacteria. Here are a few that didn't make it in, but you can see they've gotten inside the cell, and they will grow to high numbers inside your cells. The second thing that we use is called a plaque assay, and we do the same thing. We infect, but in this case, we let them go for 72 hours. And as the bacteria began to move into adjacent cells, you get these areas of clearing. So you don't have to look at this under the microscope. This is a little plate that's about 1.5 inches in diameter. And you can see that the bacteria have basically eaten their way through this monolayer. So each one of these started with an infected cell, and then the bacteria spread to adjacent cells. And this is exactly what's happening in your gut. You're getting these little ulcers formed where the bacteria are moving through your cells. And with either one of these now, we can see what happens when we treat with interferon either before infection or after infection. And again, please stop me if you have questions. If I'm getting into the weeds, perfectly happy to back up. So what we found is that if the human cells were exposed to interferon before they saw the Shigella, then they had some protection. So this is a typical, this was 6 different invasions that were done. And if they're done in the absence versus the presence of interferon, and we use 2 different type 1 interferons in this particular assay. But with beta, you can see you get about a 3-fold reduction in the number of human cells that were infected in this case, suggesting that the interferon was actually being protective. But then the student, and this was some work from Jennifer Poo, went ahead and did the plaque assay just to see what happened. And here she's using 2 different doses of bacteria just to be sure she got a range where she could see. And you get these typical small plaques after 72 hours. But if she added interferon after the cells were infected, she got these enormous plaques. So in this case, the interferon seemed to actually promote the spread of the bacteria. And here's an example under the microscope of one of these plaques. And the blue stain are the human cells. This is a stain for DNA so that you can see where the cells are. And then the bacteria are expressing a red fluorescent protein. So wherever you see red, those are clusters of bacteria. And this is the typical pattern after 72 hours. You have this initially infected cells are dying, the bacteria are moving out in a concentric circle around there. If you treated with interferon after the bacteria had gotten inside the cells, then you saw that the size of the plaques were much larger. Actually, if you measure the fluorescence, or we actually counted the number of bacteria, the number of bacteria is the same here as it is here. It's just that they're moving faster from cell to cell. So we wanted to see what was responsible for this faster move. And it was nothing about the bacteria. If you treated the cells with interferon and then isolated the bacteria, and here we're looking at known genes or proteins that the bacteria require for spread. there was no difference in those, not even a twofold change in the amount of any of those, whether or not they were growing in interferon-treated cells or untreated cells. So this wasn't some secondary effect on the bacteria. And this is now work done by my current graduate student, Ellen Gauss, kind of looked inside the cells, and the bacteria will use actin to move around in the cell, and you see these little tails. And so she had stained for actin to see if maybe there's some difference in the ability of the bacteria to move. There was no difference in the formation of tails, but what she did note is that the cells themselves, the human cells, looked very different after the interferon treatment. Instead of having these nice kind of even borders, they had these fuzzy borders, and they tended to be very closely associated with each other. And this shows it a little better in this slide. Here, the stain is a red instead of the kind of orangey stain, but this is the same thing. These are the nuclei of the human cells. This is the cytoskeleton in red. And you can see that when interferon is added, you get this spiky surface on the cells and a lot more connections between the cells. So this suggested that what interferon might be doing is altering the human cell cytoskeleton, making it a little bit easier for the bacteria to cross this border. And that is, in fact, what's happening. And I can show you more of the details. So the bacteria spread cell to cell at areas where the human cells are very tightly connected. These are called tight junctions. And at those junctions, there's a protein called E-cadherin that holds the cells together and kind of fights that cell tension that tends to pull them apart. And it's been known for some time that E-cadherin is required for Shigella to spread cell to cell. So Ellen's hypothesis is that in response to interferon, the cells began making more E-cadherin, and that this might make more places for the bacteria to move. Again, that's the same picture. So she looked at both the gene expression and the protein expression for E-cadherin. And I will say, this is after looking at about 30 other proteins. So it wasn't quite as easy and straightforward as it looks. But what she found is that the expression of the gene was up at least twofold. And this was very consistent. Every time she treated with interferon, the human cells produced more of this protein. And she could actually see it on a gel. This is a Western blot in which the human proteins are separated by electrophoresis. And then you can add an antibody against E-cadherin, and that antibody has a tag on it. So wherever the antibody lands, you can see. And so when the cell, BSA, bovine serum albumin, is just the carrier we use for the interferon. So that's our control. You can see you get expression of E-cadherin, but if you add interferon, it's up, and usually about 1.5-fold in the amount of protein being made. And this is just a normal cell protein that we use to make sure we've loaded the same amount on each one.
Shelley, I'm going to do a quick time interruption, but
yes.
Yeah.
How much longer do I have?
Well, a few minutes, and then
Okay.
we're going to have a conversation. But yeah.
So, well, let me skip the step one stuff then. Okay. So in order to show that it was actually the E-cadherin expression that was causing this increase in the bacteria spreading, we took the normal human cells and put in a gene that had extra copies of the E-cadherin gene. And she picked several of those. And you can see that when you look at the expression of E-cadherin, here's our wild type cells, which produce low levels of E-cadherin. This is the cells that now have more copies of that gene. And they produce, there's much more of the mRNA. And again, if you look in the, both in the presence and absence of interferon, these now produce very high levels of E-cadherin. And as you might expect, these cells then, when they're affected with the bacteria, produce very large plaques, even in the absence of interferon. You also see that there are fewer plaques. So the increased expression of this surface protein makes these cells more resistant to infection by Shigella, but the ones that do get in now spread significantly faster than the normal human cells. Okay. So our conclusions on this are that type 1 interferon can reduce the ability of Shigella to get inside your cells and to infect. But once infection has occurred, they spread much faster. So this suggests that viral co-infection can actually increase the severity of Shigella infection. So we'd like to see better monitoring of co-infections so that children might be treated more aggressively early if they have a viral co-infection, and that perhaps this could help avoid some of the stunting and delayed development associated with that. Okay, and then we'll stop there.
Wow, thank you. I will open for questions or comments, reactions. In a room full of, Zoom room full of non-microbiologists.
I know.
I mean, I guess so I was interested to where, I mean, right at the very end, you got to the sort of where you think this is, takes us. And so for you, it's really sort of monitoring co-infection becomes the critical piece. I mean, is there hope for other treatment, vaccination even, or strategies that would
I think,
that would be
ultimately,
upstream,
ultimately, we'd like to have a vaccine against Shigella. Fortunately, I didn't have that in my grant, sorry. But for unknown reasons, it's been very difficult to make vaccines against intestinal pathogens. And actually, the majority of your immune system is in your gut. So we tend to think about lymph nodes and at other sites, but most of it is actually in your gut because that's where you encounter most foreign agents, because you're constantly eating things. But it's been very difficult to make vaccines. I mean, you follow all the normal rules that should work, and you test it in people, and it doesn't work. So there is a lot about the immune system in the gut that's poorly understood. So we're hoping this will give some more insights into the gut immune system that could lead to better vaccines. But I think a lot of it is just really figuring out how common these co-infections are, and particularly in children who have the more severe cases. Are there particular viruses that co-infect? And we've had better success in immunizing against the viruses, and that might be one approach to this. But I think many infections turn out to be co-infections, and generally we're looking for one thing when we see an infection, and we miss a lot of those effects.
Wow.
So
go ahead.
The outbreak, and I'm from Oregon, the outbreak in Portland, I think is kind of centered locally. I don't think I've read that it's from outside other countries. And
No, you will find Shigella in the US. We had a big outbreak in Austin right after I moved here. So it's endemic. It just, you don't see much of it.
But I do think it is higher prevalence in the homeless community in Portland, I believe. Yeah.
Yes, and that's because they often don't have access to sanitation, and it's literally, you don't wash your hands thoroughly. You can have enough of the bacteria on your hands that you can get it on food. Go in any daycare center. Daycares are terrible about outbreaks. And, having had a toddler and, they go to the bathroom, then they're putting their hands on each other's faces, and they just are so unsanitary when they're that age. And you get one case in a daycare center and they all get it. So it's just, I said, it's probably the most infectious bacterium, and it's just great at spreading. So anywhere there's any kind of breakdown in sanitation, any overcrowding, military campaigns. In fact, it's, Rommel attributes Shigella and dysentery to the loss of the war in Northern Africa. So it's just so easy to spread. Now,
Okay,
see,
I'm
Deanna.
I'm nervous about Mark's time, but
Oh,
Deanna,
yes.
is it quick?
Hopefully. What about the microbiome and associations with Shigella?
Yes. So we did a project several years ago, and uh, Bacteroidetes, which is one of the major components of your normal microbiome, is protective against Shigella. And many of the areas where Shigella is at very high numbers, those people tend to have lower Bacteroidetes and higher of some other things. So there's undoubtedly connections to the microbiome and all intestinal pathogens.
Thanks. That's really interesting.
Yeah. And thank you all for enduring the nitty-gritty of.
We don't see this enough. I love it. All right, I'm going to turn over to Jay.
Thanks, Stephen, and thanks so much, Shelley. Our second speaker is J. Mark Eddy. Dr. Eddy is a clinical psychologist and prevention scientist with the Margie, Gurley, Seay Centennial Professorship in Education within the Departments of Educational Psychology and Kinesiology, as well as Health Education at UT Austin. He serves as the area chief for the school and clinical child graduate training program. In his research, he develops interventions for children and families living in stressful situations and has served as the principal investigator on multiple randomized controlled trials of programs delivered within myriad systems of care. Please welcome Dr. Eddy.
Thank you, Jay. And I just need to make sure that my audio is set up correctly because I'm going to show a video here. So just a sec. Okay. All right. Oops. There we go. Okay. Everybody see this okay?
Yes.
Okay, great. All right. So my work is primarily focused on developing and testing preventive intervention programs that focus on issues related to children and families. And I work around Texas and around the United States and in other countries around the world. But for the last 15 or 20 years, I've been doing quite a bit of work in Honduras, El Salvador, Guatemala, and Nicaragua. So I thought I'd tell you a little bit about a study that's going on in Honduras right now that's connected to that. And I'm going to kind of give an introduction to this today, and then hopefully sometime next year I could come back and speak again about findings related to this study, because we're just almost done with data collection and ready to launch into analyses on this randomized trial. But during my time in Central America and observing programs that are focused on children and families and schools, often around issues related to violence prevention, I've seen a lot of national, non-governmental organizations come and go, bring interventions in, and things flourish for a while, like the Bluebonnets, and then when the money dries up, they go away. And what's kind of unique about the program I'm going to talk about today is that could have started that way and gone that trajectory, but we made some decisions together with the people that invited us to work down there early on that has managed to keep this program around now for about 16 years. So there's lots of collaborators on this project, and some are from the UT, some are from University of Oregon, some are from a group called Child Fund International that used to be called the Children Christians uh, Fund. I don't know if you remember like those Sally Struthers commercials where you sponsor a child kind of thing. So they rebranded as Child Fund, and we've been collaborating with them. Um, so anyway, the schools that we're working in, this is a picture for one of the schools. And the place again that we're focusing on is Honduras, and particularly the capital of Honduras, Tegucigalpa. And funding for this originally came from the GIZ, which is the version of USAID in Germany. They invited us to collaborate with them, and we did for many years. And then when they decided to stop funding this, it took us a few years to get to convince everybody here in the U.S. to finally fund us, and NIH did fund us for this during the Biden administration. So what I'd like to do is just show you a real brief video clip to kind of get you thinking about what it's like in the communities where we're working. So this is about a 2-minute clip.
Prevenir es un programa regional de la Agencia Alemana de Cooperación Técnica al Desarrollo GIZ. Desde sus oficinas en Honduras, El Salvador, Guatemala y Nicaragua, Prevenir promueve la prevención de la violencia que afecta a jóvenes en Centroamérica. Con el afán de realizar un aporte a la prevención desde la educación, se construyó el proyecto Miles de Manos, que va dirigido a maestras y maestros, así como madres y padres de familia. En el corazón de Yamaranguila, Honduras, María Santiago Vázquez se desplaza varios kilómetros para asistir a los talleres de Miles de Manos. Su brazo se lo recuerda.
Yo a veces lloro al oír tantas cosas, les digo yo, hijos, oigan, tantos problemas que se oyen, ¿cómo quisiera que el país cambie? Le voy a decir, aquí nuestros hijos de nosotros todavía hasta ese extremo no han llegado, pero se dan casos ya en las comunidades de nosotros, ya sí. Eso de que, fíjate que ahorita acá nomás hubo un problema. Unos jóvenes allá en el Pericón, una comunidad aquí cerquita. Ahorita, hoy 8 días, mataron a una señora, puros hipotes. Imagínense, digo yo, y les digo yo a los hipotes, ¿será que todo esto los ayuda a nosotros para estar con nuestros hijos, aconsejándoles, diciéndoles, hijos, miren que tal cosa está mala, hijos, no está bien? Nosotros eso es lo que pedimos también, que ya las bebidas acá en nuestras comunidades no se den porque mucho problema vemos nosotros. Les sabría decir que es poco, las autoridades entre nosotros mismos aquí los cuidamos del uno al otro entre vecinos. Estoy muy agradecida y como le digo yo, me gustaría que no sea solo esta vez, que sigan con nosotros porque y hacerle más conciencia a las personas a que es bonito. Yo platico con varias amigas, les digo, vamos, bien de ver qué bonito es, vamos. Es para sobre nuestros hijos y aprende uno bastante.
Prevenir.
So the genesis of this program really started with a lot of listening, and it started with a phone call, actually a cold call I got from somebody in Guatemala. And at the time I was working in Seattle. The view out my window was of Mount Rainier. And, you know, Seattle is part of the Ring of Fire, these volcanoes that go down the West Coast. And so I got a call from somebody looking at a volcano, you know, down in Central America. So I felt we were kind of connected. And they said, hey, we'd really like to use a program that we've read about that you developed in Oregon And we've been really having a hard time getting in touch with people from the US. Nobody answers our phone. You answered the phone. We're really happy. Could we use your program in 500 schools in Central America? And I said, why would you want to do that? It wasn't developed, for use in Central America. And but I'd be happy to talk to you more. And they said, well, could you come down and meet us in Guatemala in a few weeks? And I said, well, that would be quite exciting, sure. And they said when it was, and I said, well, I'm going to be in Honduras at the time that you would like me to be there, but I could fly over from Honduras. And so at the time, I was doing some work on the island of Roatan, which for many Americans is known as kind of a cruise ship stop off the coast of Honduras. It has a history of English colonization, and people speak English on that island. And anyway, we were talking with people on Roatan about educational policy. So we went down to Roatan, and we visited schools, and we talked with families and met with principals and teachers and visited communities. And then I hopped on a flight over to Antigua, Guatemala, which is a beautiful city about 500 years old up in the mountains surrounded by volcanoes. And enjoyed just being there, and met in this old convent, which was part of a Spanish embassy there. And we convened for this meeting on prevention. And it was organized by the Germans, by GIZ. And we listened to people talk. We watched, this is a play from children where they're kind of describing what they think about the problem of youth violence in Central America. And it was really interesting to be there, and also felt very much not desired to be there. People didn't really like that we were there from the US. And unfortunately, there's kind of a bad history of US involvement, not only, I'm sure you're all aware of that, in Central America, but also particularly in Antigua. And so anyway, it was tough, and it took a while for people to see us just as people and kind of listen to what we had to bring to the table. The reason that Germany was down there in their development capacity was because at the time in 2011, it wasn't that long since a number of wars had happened, and there was really a very high murder rate in various countries. We knew that the US was linked in so many different ways with what's going on in Central America. And so we're starting with this history, this difficult kind of history that we needed to acknowledge. We were being invited in as experts, people who knew something about what they wanted to do, which was intervene with parents and teachers in schools for the benefit of children, to try to reduce the risk of their kids getting involved in youth violence and involved in gangs. And so we approach this as, yeah, we have some expertise, but we're not experts about your country. And, we're really your countries, and we're really happy to be here, but how can we have a conversation? But we were, being asked to share what we knew, what could we bring to the table. And what we tried to do was reframe the conversation from a request from them to use a program that we had developed, to us sharing information about programs that we had developed, and thinking about GIZ, the funder, as supporting the development of a new program that was written by Central Americans. One of the big tasks that we had to do was to help people shift their frame from murder prevention, which is what they wanted, to thinking about how does one get to the problem of murder? What is the lifespan development of how somebody gets involved in gangs and cartels, etc.? And so this was a lengthy conversation because they wanted to stop murder now, which we understood, but they were trying to do that through a variety of different ways. And they were asking us to help with one of those ways, but it was only one of those ways. So they were wanting us to work in schools and help them think about how to work in schools with parents and teachers and youth at a primary prevention level. But translating what we knew to their setting to be helpful to what they wanted to do was a complicated situation. It took about 6 months, but we finally did come to a new agreement with the funder, GIZ, the Germans that were in this area, but most of their employees were Central Americans, to the idea of let's develop a new program, have it be informed by existing evidence-based practices from around the world, including the US, including ones that we knew about, but ones that we didn't. And let's create a new program and develop and test that. The development team included people from GIZ, from University of Oregon, where the dean of the College of Education here, Charles Martinez, was at the time one of my close collaborators, and then me up at University of Washington, and then consultants that we encouraged GIZ to hire from each of the countries they wanted to work in Central America, as well as other consultants from the US. And so this was our original group. So there's 3 people in here from the US, and everybody else is from the 4 countries in Central America that were working. And then the guy right next to me on my left is a German who actually grew up in Argentina. And eventually the group grew and included all kinds of people from across the region. So the first thing that we focused on together was thinking about what is the theory for how they think this problem develops. And they very much were already immersed in a Bronfenbrenner social ecological kind of model. And they wanted to know what we thought of that. And we thought that's great. But then we brought in other ideas that are pertinent specifically to development of child antisocial behavior, like social interaction learning theory. And eventually we agreed on a working way to think about how this might develop in Central America with a developmental theory that was grounded in other developmental theories of antisocial behavior from the US and other countries, but very much one that they felt, yeah, that makes sense. So anyway, the idea was that along this developmental theory line on the development of these problem behaviors in children, that there are opportunities for adults to intervene all the way along the way. And how can we help equip adults, parents, caregivers, other caregivers, and teachers to help divert a child's path if they're on their way to developing violent behaviors, delinquent behaviors. The group that we were working with in these 4 countries, we would meet with them regularly 4 times a year, and then have many, many calls, not on Zoom, but on prior iterations of things like Zoom. And we tried to come to an agreement with the group about what is it we're going to focus on. Feelings, thoughts, behaviors, what is it we're going to do? And eventually we got to this idea that let's try to focus on behaviors. What are things we actually could do as parents and teachers that might make a difference? The programs that they were really interested in really were around development of skills, which is why focusing on behaviors seemed like a good idea. So this is an example. And eventually we did come up with a variety of ways to kind of illustrate what was being thought about. And this is an example. But a key thing is that this particular program, which eventually was called Miles de Manos, or Thousands of Hands, was just part of this broader effort that the German government, through their International Development Corporation, was funding. And already when we were invited in, they had developed community level prevention committees in a variety of communities around these 4 countries in Central America, community policing support, and adult employment assistance. And so they were asking us in for the fourth leg of the table, which is school-based and family-based violence prevention activities. So the intervention that was developed, eventually the idea, and again, this is coming from the people from Central America who were interacting with us, but they drove the writing of this curriculum. But the main idea is to unite and strengthen families and educators in their efforts to help children and youth effectively deal with challenges in their lives, and then specifically foster caregivers and educators' capacities and skills that can help them best support their children and youth. So there are three components to the program that was developed, and this took about 3 years of work and writing, going out into communities and testing it out, coming back. And the first community we worked in was illustrated in that video I showed you. Was way out in a rural area in the mountains. And anyway, there's three components. One is for parents and other caregivers, one is for educators, and one is Bridge, which brings the parents and educators together. And the idea is to provide skills for teachers, training and skills and support for the use of skills that help teachers during the school day. Same for parents and caregivers outside of school, and then bring them together to talk to each other, which was really important because parents and teachers in the communities that we were working in often didn't really talk to each other very much, and certainly never had meetings where there were multiple teachers and parents together to talk about issues related to children and kind of try to get on the same page. So that was kind of a novel part of this intervention. In the end, the program focuses on helping develop effective communication skills that are between parents and their kids, between parents and teachers, and between teachers and their students, and then other aspects of communication that support effective communication, like emotion management and etc. And the process of the intervention that was developed is delivering the content in multiple sessions in gatherings that aren't too big, where there's food and childcare and social support, and to provide an overview of several skills to teach that day, having an opportunity to practice skills in the session, and then encouraging home practice. And so in the end, these different curricula were developed: a family component, the educator component, and the bridge component. And it's really a very interactive kind of way to do things. The idea is that the people running the program are facilitators, not lecturers. Would never do something like I'm doing today, just talk, talk, talk. Don't do that. And practice is really important. And so in the end, this sequence was developed for starting with the bridge and getting a common frame for parents and teachers, and breaking up and having teachers meet, parents meet, and then coming back together periodically. And so anyway, this is what kind of a typical school that we're working in now looks like. And this was informed by evidence-based practices that many that we had developed on our team, but other ones that they were interested in. And this is an example of our team working together in San Salvador. And so in the end, we got this program developed and developed this supervision and support chain for how do we support the facilitators that are delivering the program. And ultimately, this program that was developed was implemented pretty widely across 4 countries, eventually to over 200,000 students, but this was an early thing. And it took us a few years to get a randomized controlled trial funded. The Germans weren't interested in doing that. And then during the first Trump administration, we went through peer review, got really good scores, but they wouldn't fund it because they didn't want to fund things in Central America. But finally, when the Biden administration came in, we put it in again, it got funded. And we are working in Tegucigalpa in neighborhoods that are kind of at risk for gang involvement, but haven't gotten hot. So the red neighborhoods here are the hot neighborhoods. They happen to be in between the downtown and the new airport, which is about an hour and a half out. So you have to go through that neighborhood if you're ever there. But the trial that we're doing is focused on third, fourth, and fifth grade students in 30 primary schools. And we're doing randomization at the school level. We're doing 3 assessments, a pre, a post, and a follow-up that happens about a year later. And what happened when we finally got funded? Well, COVID came in and all the schools in Honduras promptly shut down. And they were closed, in fact, for 2 years. So we were not able to do any of this work. And so then when they finally got started up again, we got going. But it's been challenging to do this trial, not only because of COVID, but also multiple presidential elections where the entire middle management that we're working with will go away. And we have had IRB review both in UT Austin, but also we wanted an IRB in Honduras. So the National University Medical School there has been providing that for us. And so we've got 2 IRBs to go on. But this is us on a recent trip visiting. So my idea was to give you a little background on this, and hopefully I can come back sometime next year and tell you what the results are of this trial. So any questions? Well, it's been fun.
Wow.
I feel like, the key moment here, I think, was to not accept the original invitation to just use our program, but to actually encourage a new program to be developed. So that's the thing I think I'm most proud about on this one, is that we encourage them to do something new and to have it be authored by them. And the thing is that we've been doing this work since 2011, and this program still is around in all of these countries. It hasn't disappeared like these other programs because it's it was developed by writers from Nicaragua, Honduras, El Salvador, and Guatemala. And people know that it was created in Spanish, not in English. And it's been a real honor to be able to do this trial and create a new iteration of the program. And anyway, so
Yeah.
I'm excited
Well,
about
and
it.
my thought was that there was like an entire parallel, like whispering in your ear part of the whole story about like, how did you pull this off for that long and sustain it, and ups and downs of funding and all that. I mean, that's remarkable too. Like a good program can like take a life of its own, but somehow you've also been engaged enough to like get to the point where you're doing a trial of it. I mean,
Yeah, it's exciting. I'll be down there in about a month, and then and again in a couple months later. The really sad thing at the moment is that we were on the verge of negotiating some things to keep it going after we're gone that were related to a $85 million grant that RTI had from USAID, but it is gone now. So,
right. Of
so
course.
that's gone. But we are trying to see, is there any way to keep this going? We have started with the principle, by the way, of having the resources that were developed be available for free rather than need to be purchased. So all the manuals are available, and there isn't like a cost cliff you have
Yeah.
to go get it. So we did negotiate some things up front to say, let's not create something that we're going to sell. Let's have it be available. So that's been helpful. But of course, it has its own challenges. But anyway.
I think your project was an ex excellent example in sustainability, Mark. Just getting key
Yeah.
stakeholders involved, and keeping it
Well,
going.
thank you. And thank you both for this terrific range from the organism to the community all over the world. So it's really cool. Thanks,
Yeah,
Shelley.
Shelley, thank you so much. That was such an interesting presentation. And
And thank you for letting me practice my Spanish.
Oh yeah, that was cool. Yeah, thanks for doing that. All right, well, good to see y'all.
Good to
Until
see
next
you.
time.
Thanks for the invitation. Yeah, take care. Bye-bye.